compare that to a ladder. So a ladder, when you go to a lab company that sells a DNA ladder, they're giving you a tube of known size sequences in that tube. So there's a sequence that's 100 nucleotides, a sequence that's 200, one that's 300, one that's 4, 5, 6, 7, all the way to about 1,000 usually. So you'll have these bands that go from 1,000, 900, 800, 700, 600, 400, right, so on. That will allow you to judge how large the bands in your sample actually are. So if you're looking for a fragment that you've amplified around 600 base pairs, there should be a band right next to the ladder where it says 600 on the ladder. So that will give you an indication of if your PCR worked. If it worked, then you can go on to sequencing. So the DNA sequencing, there's lots of technologies out there. There's 454 sequencing, Illumina, Smart Sequencer, Ion Torrent. All of those are modern sequencing technologies. Sanger is an old one that was developed in the 80s. Really nobody uses that anymore. All of the new technologies, they use this very high-powered camera, right? This camera is the bulk of the expense of the machine. So when you're spending a couple $100,000 on a DNA sequencer, 190 something of that is the camera. And this is a camera that you put on, a telescope satellite or something, Hubble, right? It's a very powerful camera that can detect the little flashes of light when a new nucleotide is incorporated into the growing chain. So you're going to have this plate that's about the size of your phones that's going to have over 100,000 little wells. And each well is going to have a DNA sequence that's about 1,000 to 5,000 base pairs long inside that well. When you add in nucleotides that are different colors, usually, you're going to have fluorescent tags to each. So A is green, G is blue, C is orange, what have you, right? You're going to have these different flashes of light that the camera is going to pick up. So the camera understands where each flash of light originated from each well,